So now let us take a look at the sexual reproduction. In Ectocarpus, the sexual reproduction is both isogamous and anisogamous type. That is isogamous, which involve gametes of similar morphology. Okay, the gametes they are similar in morphology. That is similar shape and sizes. And anisogamy, that is they differ in size as well and or in form. Okay, so they may be differ in size and in their form, these two gametes. So that type of sexual reproduction is anisogamous, and anisogamy is very common in Ectocarpus species, and it may be of two types, that is morphological and physiological. Morphological anisogamy, where the gametes they are dissimilar in their sizes, and physiological they are dissimilar in their physiological function. Okay, where we will see that one gamete is it has motility. Okay, it can move, whereas the other one it cannot, it is immotile. So they show physiological dissimilar physiological function. So that is why in Ectocarpus the anisogamy sexual reproduction it may be of two types, that is morphological and physiological, and these gametes they are produced inside the plurilocular gametanja. Okay, as you can see here in this figure, these are the plurilocular gametanja, where and they are developed on the haploid plan. Now, the development of these plurilocular gametangum, they are very much similar to the plurilocular sporangia. Remember, just now we have studied the development, the formation of these plurilocular sporangium. Okay, these sporangium in asexual reproduction, they will form the zoospores. Okay, the diploid zoospores will form on these plurilocular sporangia, whereas in sexual reproduction we will see the formation of these plurilocular gametanja, and they, as you can see in this figure, they are large, elongated, and they are sessile or short stalk and multicellular structure. Okay, you can see many cells. That is why morphologically both the plurilocular gametanja and the plurilocular sporangia they are alike. You can see in this figure also these gametanja they are very much similar to the plurilocular sporangia. Now the plurilocular gametanja they produce haploid gametes. But on the other hand, just now we have studied that these plurilocular sporangia they produce diploid zoospores. They produce diploid zoospores, whereas these gametanja they form haploid gametes. Okay, so that is the difference. Now, though both are morphologically more or less alike, now also one more thing which is different here is that these gametes which are produced, these haploid gametes, they are slightly smaller in size. Okay, now let me show you. These gametes they are similar to the zoospores, but then they are much smaller in size as compared to the zoospores. Okay, these zoospores which are formed in plurilocular sporangium, and the development of plurilocular gametanja is alike with the development of the plurilocular sporangia. So I will not explain again the development since they are very much alike. And the gametes also they are liberated from these gametanja in a similar manner. Okay, following the same procedure as that of zoospore liberation. You can see in figure B. Okay, they are released in the same way like the zoospore. So This is the formation of the plurilocular gametanja. Now we will take a look at fertilization. So next here in figure C, you can see what happened during fertilization. Majority of the species of this Ectocarpus, they show physiological anisogamy. Okay, but morphological anisogamy is observed in some species only, such as Ectocarpus secundus. Now, in physiological anisogamy, as I said, that it means the sexual reproduction, okay, which takes place where both the uniting gametes, the male and the female gametes, they are morphologically similar. But why it is referred to as physiological anisogamy? Because they are differ in their physiological function. Now, whereas in morphological anisogamy, they are differ morphologically, where the female gamete is usually larger in size than the male gamete. So majority of the species, just remember, they show physiological anisogamy. Now what happened during fertilization? Many male gametes, as you can see here, they encircle the female gametes. You can see these are all the male gametes here. Okay, they encircle the female gamete. This one is the female gamete. So they encircle the female gamete, and as you can see, they get entangled by the anterior large flagellum. You see all these, they are biflagellate, right? One is shorter, one is longer, the flagella. Now they get attached, they get entangled with the female gamete through their anterior large flagellum. So this stage here is referred to as the clump formation. Okay, so this is referred to as the clump formation, where many male gametes, they encircle the female gamete and they get entangled to it by the anterior large flagellum. Now out of these many male gametes, actually only one male gamete fuses with the female gamete. Okay, only one will fuse with the female gamete. Now in figure D, you can see where the male and the female gamete, they fuse together, and the remaining gametes go astray and they gradually get destroyed. Now the uniting gametes, then they form zygote through plasmogamy and karyogamy. Plasmogamy, that is the fusion of the protoplasm, and karyogamy, that is the fusion of the two haploid nucleus. We know that the male and the female gamete, they are haploid, right? They are haploid, so when they fuse together, they will give rise to a diploid zygote. So here in figure F, we see this is the diploid, the formation of the diploid zygote. Now the germination of zygote, this zygote, it undergoes germination without any reduction division. Okay, that is the sexual reproduction, the germination of zygote, it takes place without any reduction division. That is, it remains as diploid zygote. It will not undergo any reduction division. That is, it will not reduce from a diploid to haploid. Now on germination, this zygote, it will develop into a sporophytic diploid plant. Okay, so this sporophytic diploid plant, it will then again develop unilocular and plurilocular sporangia. So this is how the fertilization and germination of zygote occurs in the ecto. So at the time of germination, the diploid zygospore, nucleus, it divides mitotically. Okay, hence it will give rise to a diploid thallus. Now here taking a look at the if we take a look at the life cycle, Okay, the alteration of generations in Ectocarpus, we see that the sexual thalli of Ectocarpus, they are dioecious and they are also haploid. They produce only plurilocular gametanja. You can see here in this figure. They produce only the plurilocular gametanja and not the unilocular gametanja. So the iso or anisogametes, they fuse to form diploid zygote. Just now we have studied that, we have discussed that they fuse to form, to give rise to the diploid zygote. You can see here the zygote is diploid, and at the time of germination the diploid nucleus of the zygote, it divides only mitotically, and thus the zygote, it forms the diploid sporophytic thallus. Such thalli, they bear both unilocular and plurilocular sporangia. You can see here they bear both unilocular and plurilocular sporangia, and the zoospore form in plurilocular sporangia, they are diploid and on germination give rise to diploid sporophytic thalli. Also we had already discussed that the the zoospores formed by these plurilocular sporangia, you can see this one, the plurilocular sporangia, they give rise to a diploid zoospore, right? The diploid zoospore, and on germination they give rise to the diploid sporophytic thalli, whereas the haploid zoospores, which are formed in unilocular sporangia, on germination they form haploid gametophytic thalli. Okay, so these zoospores which are formed from the unilocular sporangium, they give rise to the haploid gametophytic thalli. So what do we see here in the life cycle of Ectocarpus? Both the gametophytic as well as the sporophytic generation, they are morphologically similar. Thus, such a life cycle is referred to as isomorphic. So just remember that a life cycle of Ectocarpus is isomorphic.